Okay, hello everyone. Welcome to today's session where we are going to discuss a question which has appeared in GATE 2017 question paper in Excel code, and this question is question number 35 from biochemistry section. So the question says that a newly isolated circular plasmid gave two bands of 3.2 and 3 kb on digestion with EcoR1 and two bands of 5 kb and 1.2 kb on digestion with BAMH1. And double digestion with EcoR1 and BAMH1 yielded four bands of 2.6 kb, 2.4 kb, 0.8 kb and 0.4 kb. Digestion with SAL1 leads to disruption of ampicillin resistance gene cassette. The correct restriction map is. So these are the four options given, and it has been asked to us to tell us according to the questions which one is the correct map. Okay, so before starting, so let's talk about this restriction enzymes. Okay, so in this question we are provided with four different circular DNA. Okay, circular DNA which are nothing but plasmids, or we can also call it vectors. Okay, so these are the circular double stranded DNA. Okay, circular double stranded DNA which are present inside the bacteria. Okay, so these circular DNA or the plasmids are very useful and involved in recombinant DNA technology. So recombinant DNA technology is a technique which is being used nowadays in laboratory very frequently to create the modifications in the DNA, or to create certain type of construct which will help the researchers in their experiments. So this circular DNA plasmids or vectors, whichever way we say, so these vectors have certain features. For example, it should have a particular origin of replication, so that whenever you put this vector inside any particular host cell, it can replicate within it. So this is an origin of replication site. Along with that, it should have a promoter sequence, okay, which will help to transcribe the specific gene product which is cloned in it, which is put or inserted in this vector for our main purpose, for our desired purpose. Okay, so gene, so whenever the promoter is active, it will start producing that gene, and that gene product can be used, inside that host cell for our specific purpose. Suppose I am supposed to put this particular vector in a bacterial cell. I clone a certain, or I insert a certain gene inside it, which will be produced inside the bacteria. So this entire system which is taking place inside this bacterial cell. Now the bacterial cell, which is a host cell, does not produces this particular or gene of interest. So that is why this gene we are putting inside a vector, in frame of this vector along with a promoter, so that this vector will be this promoter will actively transcribe this gene, and this gene keep on producing the protein inside a bacterial cell. This gene could be anything, any prokaryotic protein. Generally it is an eukaryotic protein which we are trying to express inside the bacterial host cells, and this protein we can use according to the experiments that is designed. Okay, so these vectors are very specific, and because it has origin of replication, every time the bacterial cell divides, this plasmid also divides, and that also get transferred to the daughter cells. So this copy of the plasmid remain maintains throughout the bacterial uh, uh, divisions. Okay, and along with these two things, the bacteria also have I would say antibiotic resistance gene. Okay, so antibiotic resistance gene, it could be any gene that provides resistance against certain antibiotics. So antibiotics are for specific for bacteria that kills the bacteria. Okay, so there are like one bacteria, there are two different bacteria. So one bacterial cell which will harbor our vector. Okay, so suppose this vector I am transforming inside the bacteria, and other cell which does not have this vector. So this uh, like suppose this is bacterial cells, uh, these bacterial cells are sensitive to ampicillin, which is an antibiotic. Sensitive to ampicillin, which is an antibiotic. But when we are putting this vector inside this bacterial cell which is harboring the ampicillin resistance gene, that means it will produce such a gene product that can overcome the effect of ampicillin. So this cell which does not have any ampicillin resistant gene, or which does not have this vector so that the ampicillin resistant gene can be produced, it won't survive when we give ampicillin. Okay, ampicillin containing media. When we give ampicillin containing media, out of these two bacterial cells, the cell which does not harbor this vector will not survive, but the other cell which harbors this vector will survive. So this uh, is used for the selection of bacterial cells harboring the vector, harboring the plasmid. Okay, so these plasmids have any particular antibiotic resistance genes. So the cells which are not resistant, now after taking up the plasmid, they become resistant to those antibiotics, and the other cell which have not taken the plasmid, they won't survive. That's how we are able to select the particular cell which have taken our desired plasmid. Okay, so ampicillin resistance gene. Other than that, the bacteria, this vector or plasmid which also harbors a small site which is called as MCS, or we can call it as multiple cloning site. Okay, multiple cloning site, MCS. Okay, MCS is used for clone a particular gene of interest. Like suppose this gene is being cloned, so it is generally being cloned with the help of uh, restriction digestions. Okay, so in this MCS we have several restriction sites. Suppose BAMH1, SAL1, EQUAL1, NOT1, KPN1, SLA1. So all these which I am writing here, these are different restriction enzymes. So what is a restriction enzyme? Sites. Okay, so restriction enzymes are specific protein or enzymes which are produced inside the bacteria to cleave a particular foreign DNA, generally viral DNAs. Okay, so these restriction enzyme makes cut at specific sequences. For each restriction enzyme, the sequence is unique. Okay, each restriction enzyme has a unique detection or identification sequence. Okay, so that's how these restriction enzyme identify the target sequence. Okay, so as we have different restriction enzyme, we have different restriction sites. Suppose for BAMH1, this is a restriction enzyme. Okay, and the restriction site which it recognizes is, so these are generally, restriction sites are generally 6 to 8 base pairs long double stranded DNA sequences. Okay, and these enzymes recognize these sites specifically and make a cut. And after making a cut, it will produce a fragment which will look like this. Other fragment will be, so these are palindromic sequence generally. Okay, so these two sequences will get produced. Okay, so these restriction enzymes are majorly of two types, so type 1, type 2. So if I'll talk about type 1 which recognize a particular target sequence but make a cut a little far away from that sequence, and type 2 restriction enzymes which recognize the sequence and make a cut also on those sequence. So these BAMH1, EcoR1, NOT1, KP1, those come under the type 2 restriction enzymes. Okay, so type 2, that means make cut in the recognition sequences only. Recognition sequences. Okay, so these will cut the DNA. So suppose if I have a circular plasmid, okay, this is my circular plasmid, and here somewhere I have the sequence which can be identified by BAMH1 enzyme. So here I have this sequence. Okay, so when I add BAMH1 enzyme, so this enzyme will make a cut here, and now after the digestion or after the restriction digestion, it will be something like this. It will remain, it will be open. Okay, so from circular now it will become a linear plasmid which have the restrictions ends generated. Okay, here BAMH1 restriction sites generated at the ends. Okay, and if we have two restriction sites of BAMH1, suppose here also BAMH1 site, here also BAMH1 site, then it will after digestion again this portion will come out and this portion will come out. So this circular plasmid now will give you two fragments of different sizes. Okay, so that is how these restriction sites cut the DNA and digest it. Okay, so now if we'll see our question here. So a newly isolated circular plasmid. So all four are circular plasmids. So gave two bands of 3.2 and 3 kb on digestion with EcoR1. So this 3.2 and 3 kb is the sizes of the DNA. Okay, we generally express the sizes of the DNA in the kilobase pairs or base pairs. Okay, so on digestion with EcoR1, it is giving, okay, so if this plasmid is getting digested with EcoR1, it is giving you a two bands which is of size 3.2 and 3 kb. Okay, two bands. And then uh, and two bands, and when BAMH1 digestion, okay, so it is giving 5 kb band and 1.2 kb band. These two bands. Okay, and when digested with both, then it is giving bands of 4 bands. 2.6 kb, 2.4 kb, 0.8 kb, and 0.4 kb. This is what is given in the question. Okay, and the digestion with salmon lead to disruption of ampicillin resistance gene cassette. Okay, so the correct restriction map is, so if I'll tell you very simply, this question can be solved, no need to look all this data very easily. Very first thing which you are supposed to see is where is the salmon restriction site is located. They are saying that salmon restriction site or salmon digestion is disrupting the ampicillin resistance gene cassette. That means the salmon restriction site is present in between this ampicillin resistant gene cassette. That's why it is getting disrupted upon restriction digestion with salmon. So if we'll see all four, so this first one, this is the ampicillin resistant cassette, and salmon restriction site is away from it, and salmon is such an enzyme which makes a cut at the recognition sequence itself. Okay, so it is type 2 restriction enzyme. So if the site is situated here, the cut will also be made here, but because of this cut, the ampicillin resistant gene cassette won't be disrupted. So this is a wrong option. The next one, there also salmon site is away from the ampicillin resistant gene cassette, so this is also a wrong option. This option, here also we can see that salmon is away from the ampicillin resistant gene cassette. This is also a wrong option. So the fourth option, if we'll see here in the ampicillin resistant gene cassette, this portion which is being shown, the salmon site is present there only in between the ampicillin. So now after digesting with salmon, this will become linearized, and ampicillin resistant cassette will be disrupted. It will be disrupted. It will be divided into half upon salmon digestion. Okay, so if we will not see all these sequences also, or then also for sure we can say this is our correct option because of the presence of salmon site in the ampicillin resistance cassette. Okay, now let's cross check all these three data which is given. Let's match them, and if it is in that eco r1, okay, so eco r1 is cutting, making a, it has two restriction sites, then only making, giving us two bands. So if we'll divide it, 3.2 kb and 3 kb. Okay, and BAMH1, which is also making two cuts, so this portion and the other portion, bigger portion, which is 5 kb and 1.2 kb, which we are assuming because the exact distances are not given to us. So according to this, we are just assuming this is the scene, and if this is the scene upon, so BAMH1 is, it has given us like 5 kb and 1.2 kb, and then again eco r1 is cutting this 5 kb sequence, dividing it into two, which we can see here, these two, 2.6 kb and 1.4 kb, and this 1.2 kb is divided into these two. Okay, 0.8 kb and 0.4 kb. Okay, so just this is a simple way to solve this question. Very simple. Just look for the salmon restriction site where it is present, and you should know that salmon is a type 2 restriction enzyme which makes a cut at the recognition site itself, not away from it. So if the salmon site is present in the ampicillin resistant cassette, that map is the correct map according to the question. Okay, so this is the answer of the question. This is my NPTEL reference. Thank you for watching the video.